All right, well, thank you, Christina. Thank you, Tracy, for the invitation to drive down the road from Seattle to Renton here and speak to you all. For those of you that are not from here, I hope you appreciate that it's always like this in Seattle. It's always 80 degrees and sunny. So I want to tell you that I'm not a pediatric neurologist. I'm an adult epilepsy specialist. I work at the University of Washington Regional Epilepsy Center. So we're a tertiary care referral center. I have 8 other full-time epileptologists who work with me. And we see patients who are primarily referred to us from other community neurologists who are difficult to treat. And some of them are sent to us to treat difficult to treat epilepsy with medical management. Some are sent to us for consideration of surgical treatment. Generally, I don't have a lot of easy patients in my practice. I have a few, and I'm always happy about that. But most of our patients are what we would call medically refractory. What I'm going to talk to you today about is a little bit of a research project that we have been, a research effort that we have been undergoing for the last 10 years or so to understand what can we do for people with medically refractory epilepsy that's in the here and now. What can we do with the medications that are available to us? And can we acquire any kind of evidence that will drive our decision making? And I call this approach, I didn't invent this term, but it's a trendy term, so I used it, big data, because we're relying on the experiences of many people and aggregating them and trying to look for a pattern. So what I'm going to tell you is not specific to LGS. I have LGS patients in my practice. They're adults. They become adults. I have adults in my practice with LGS. But I'm going to tell you about refractory epilepsy as the whole, because that's the problem that we're trying to attack. And I'm hoping that some of this is going to be relevant specifically to LGS. And as Christina said, we're generously funded with some of your money that you helped raise to do a seed project in this regard. And at the end of this, I'm going to tell you how each and every one of you can contribute, not with money, but with your efforts and what is happening with your children to further our research. Okay. There's a brief disclosure. Okay. So the problem of refractory epilepsy. We say that epilepsy is about affecting in the US about 3 million people. About 1/3 of those people are we call medically refractory, which means they've been exposed to 1, 2, 3 drugs without achieving seizure freedom. As an epileptologist, that's always my goal, is seizure freedom. And so to some extent we say, well, we have failed when we haven't achieved that. So that gives you about 1 million people in the US who have poorly controlled epilepsy. And as you know, if you have poorly controlled epilepsy, even if it's 1 seizure every 6 months, as an adult, you can't drive. Maybe nobody will hire you. You're probably not going to have all the full benefits of education, all those life milestones that you want. So that, in my mind, that is the problem that we are trying to attack. Now you saw this slide. This is a slightly updated slide from the one that Tracy just showed you, but it just shows you the tremendous productivity of the pharmaceutical industry over decades in cranking out new medications. Been very helpful for us neurologists because it gives us a lot of different choices, and not choices so much that are one more effective than the other, because honestly, that's not really what has been our experience. The choices are side effects, and matching the side effect profile to our individual patients to make them more tolerable to them in day-to-day use. The unfortunate truth is that despite all this research productivity and drug development productivity, none of these drugs in refractory epilepsy as a whole has been a game changer. We're still waiting for that. So still, despite the decades of drug development, still, as in the 1970s, as in the 1990s, about a third of all patients with refractory epilepsy, a third of all patients with epilepsy remain medically refractory. Maybe nibbling away at that as we introduce each drug, but no game changers yet. So with all these drugs, when you go to your clinician and you say, how should we attack this problem, whether it's LGS, whether it's temporal lobe epilepsy, what have you. What's the algorithm? Well, there's not really an algorithm. How many combinations could we construct out of all these medications? Well, if we just took the top 12 of them and combined them in different ways, 298. So how do you make sense of 298 possible ways to treat epilepsy when we don't really know? Now, let me tell you a little bit different story in a different disease condition. Imagine, God forbid, that one of your loved ones had breast cancer. If you had breast cancer, here's how your clinician here at Seattle Cancer Care Alliance or anywhere else would approach it. First, they would get a tissue diagnosis. They would look for biomarkers on the tissue. Then they would do a genomic screen of that tissue to find out the chance that it would recur and how aggressive the tumor is. Then, and this is the important part, they would have access to dozens upon dozens of large, well-constructed, NIH-funded clinical trials that would have tried drug A versus drug B versus drug C, drug A plus B versus A, drug A for 1 year, drug A for 2 years. All that information, beautiful evidence-based information, and you would go down a treatment path for exactly your condition. We don't yet have that in epilepsy at this moment in time. You're going to hear talks about how things are getting there, particularly for childhood catastrophic epilepsies. But for the average person who walks into my clinic, we don't have that kind of data. So the research data I'm going to show you now is our attempt to be in the here and now and say, instead of conducting many large prospective trials to answer this question, what kind of data can we aggregate with the drugs we have in hand and with patients who've already been treated to see is there any evidence for something that would work better for our patients? That's what we're trying to do. So the way we hit upon doing this was very serendipitous. Myself and my colleagues are consultants to several Washington state institutions for severely developmentally disabled adults. These are people who are in institutions. They don't walk, they don't talk by and large, and about half of them have refractory epilepsy. And we would go once a month to these places. One is up north in Shoreline, one is just in the shadow of Mount Rainier. And we would go there and visit with the patients and clinicians, and we would see them one after another and make recommendations about their treatment of epilepsy. And it turned out that every time we would go, the pharmacists there would present us with these lovely coded graphs of how many seizures they had had over the preceding several months that had been tracked by the nursing personnel there and plugged into Excel spreadsheets. This is very helpful for us. Look at the ups and downs and how the drugs changed and make recommendations. And after having done this for about 8 years or 10 years or something, as I was driving home from the clinic one day, it suddenly occurred to me, like, wow, this stuff is all in Excel spreadsheets. And it turned out to be 30 years of Excel spreadsheets. I was like, there's got to be something in that information that we could look at. And so we wrote a IRB to get the data from the state of Washington, and we did a little bit of analysis. So I'm going to show you what we found in that initial analysis. So the question was basically, we looked at the most frequently used drugs, and we just counted seizures. And these were seizures observed by nursing personnel. They were mostly convulsive seizures, mostly big seizures. And the simple question we asked is, within each individual patient, and each individual patient had been on multiple drug regimens, was there anything in that individual patient that worked better than anything else? So we were just comparing drug A or regimen A versus regimen B, et cetera. And doing that within each patient. The reason to do that within each patient is because there's such individual variability amongst people with epilepsy, especially these severe epilepsies. So we wanted to normalize for all the differences in their seizure frequency by making our comparisons within each person. And then the simple question we just asked is and we asked a number of questions. I'm just going to kind of give you the bottom line. But the question we asked is there anything that works better compared to anything else that each person had been on? Okay. This is a table that I don't know how readable this is, but it kind of gives you an idea of the sort of folks that we were looking at. I'm going to highlight the diagnoses of their conditions, and that's that middle group there. So genetic was about 10% had an identifiable genetic cause. LGS was in there about 8%, perinatal hypoxic. Still about 60% there was no specific diagnosis. And this is because many of these people, these are adults, they had been in there for decades, and a lot of this predated some of our modern methods of diagnosis. Here's the data that we had to look at, and I'll just mention the top line. For our average patient, we published this as 2 studies, and this was the second study that we published a couple years ago. The average patient, we had 17 years of continuous data. So contrast that with a typical clinical trial that lasts 12 weeks. We had a lot of data, and this is what made this study, in my mind, work, is because we could follow patients over years and years and years and years and see how things were working, not just over weeks. Here were the drugs that they were most commonly used on, and I've used the standard acronyms for the drugs, but you can see that patients were disproportionately, they were exposed to everything under the sun in reality, but here's the top 12 or so drugs: lamotrigine, valproate, carbamazepine, phenytoin, topiramate. They've really been exposed to everything under the sun, but all sorts of different things, and exposed to these drugs in no protocol or pattern, because this was under, over years of we clinicians coming to clinic, making suggestions according to what we thought would work best. No rhyme or reason, trial and error. So in reality, we covered a lot of different possible combinations of therapies, but no rhyme or reason to anything we chose. So when we crunched all the numbers, here's the conclusion we came up with. First of all, so this is a table that shows in order of the number of different patients exposed to each drug regimen, and you can see about 25 of them there from top to bottom. Carbamazepine and monotherapy being exposed to the most number of patients, and going down, and you can see 2 drug combinations, 3 drug combinations. And the table, the column there that says average % decrease or increase in seizures, you can see that on the average, some treatments made people a little bit better, some it made on the average a little bit worse. And all of that, with except for one treatment, was completely lost in the wash. So everything else was just really nothing, there were hints that maybe this was a little bit better, that was a little bit better, but not achieving, what we would call statistical significance, except for one. And the one drug combination that made the difference in this group of patients of very heterogeneous epilepsy syndromes was the drug lamotrigine, otherwise known as Lamictal, plus valproate, otherwise known as Depakote. And the 2 of those together, which is highlighted in yellow, on average reduced people's seizure frequency 50% compared to anything else they had been on, or the average of everything else they had been on. And for those of you who are into statistics, the p-value of that was 0.00001. It was a large or a small number. So that was interesting to us because it really stood out, and it was a little bit, some people would say it was expected, but to us, the way that stood out was very surprising. The interesting thing about this, in this patient group, as I said, they were very heterogeneous in the causes of their epilepsy. And this drug combination worked equally well in those who had generalized epilepsy, which we would consider LGS to be more in that category, but also in those who had focal epilepsy, like those with brain trauma or very focal brain lesions. It worked equally well. It seemed to us that the effects of these 2 drugs together was synergistic because it was better than either one of them did by themselves, or if you kind of mentally added the effects of either one of those 2 together, this was far better. And for reasons that I won't really go into, it appeared to represent an effect where the 2 drugs were doing something synergistic at the level of the neuron, not because of the way they were metabolized or the way they affected each other's blood levels. It was some kind of a magical pharmacodynamic synergy, which is the kind of thing that we always like to talk about achieving, but rarely do. Now, how to put 50% decrease in seizure frequency into perspective? That's not curing epilepsy. I mean, I think we'd all agree it's a good thing, but it's not necessarily curing it. And there were a few patients in our cohort who became seizure-free, but most did not become seizure-free. But when I think about it in the context of should we consider a surgical intervention for some of our refractory patients, in cases where we're not thinking about, say, doing a resective surgery, but we're thinking about placing a device like a vagal nerve stimulator or a responsive neurostimulator, just comparing the published first-year efficacy of those surgical studies, this would appear to be better. So I would say in our center, I've talked about this a few times, and I practice with some very smart, very skeptical colleagues who have heard me talk about this, but have given me all their caveats all the years. But in their own practices, I've seen them adopt this more and more as kind of a standard of practice for our refractory patients before they go to something surgical. Now, there are issues with this study that we conducted. The most obvious one is we were treating these severely developmentally disabled patients. And you could say also this was a very specialized practice setting. There were a small number of physicians, maybe a dozen or 15 over the years, who were making treatment decisions. And you could say maybe there was an artifact of people kind of got into a practice pattern and they did certain things that maybe weren't replicated in the broad community. So the next phase of our study, which was funded by the Lennox-Gastaut Foundation with your generous support with the seed grant in 2017, was to ask the question, could we find any applicability of this to a broader patient population in the community? So the question is, how do we find that kind of data? At Fircrest and Rainier, we had institutionalized or practices institutionalized where the pharmacists and the nurses and everybody were recording seizures diligently. Medications were given with complete, precision. There was no non-adherence to medications. The data was perfect in terms of the way it was collected. So we said, how can we find that in the community? Well, we can't. But our idea was then to go and use what I would call a crowdsourced data source. And the tool that we turned to initially was Seizure Tracker. All right, any of you folks using Seizure Tracker? How many? Can I see your hand? I'm just curious. A few. Okay, it's good sprinkling. Okay. So this was a software tool developed by a guy named Rob Moss and his wife. They have a child with severe epilepsy, and they just wanted something where they could record their child's seizures and how they're giving the medications, and do it from their phone or their computer and have it exist online, and then they could go to their physician and show them the data. And with the idea that, just like we at Fircrest were able to base our decision-making on graphs and real data, so much more helpful for the physicians when you can give them real numbers. And so this tool has been out there for a few years now, and they've signed up a number of people, 25,000 users over time. And so I and other people have gone to Rob and said, let's have your data and see what we can find. And initially, he was very skeptical about that because of privacy issues. But gradually, he came around to the idea that this would be a useful tool for the community. And so long story short, he came up with a method to completely anonymize the data and make it available to researchers. And there have been several publications that have been put out there using this data, mostly from an NIH group with Bill Theodore and Dan Golden-Holtz, but that's just some of the publications. So we obtained in 2017, as part of this seed grant study, we obtained the data set. And let me just show you what it looks like. So I mentioned there's about 25,000 users in it. And Rob will tell you that there are 1 million seizures documented in that data set. So hence the sort of big data number. It's a different kettle of fish than our initial data set. And the reason is that even though 25,000 very well-intentioned people signed up to use this data set. When you start drilling down to see what is going to be usable for our kind of study, which is a very specific kind of study, you find that, well, okay, only 15,000 people, 25,000 people signed up, but only 15,000 people ever put any data in. And then how many kept at it for a year? Well, only about 1,800. And then how many people kept at it for a year and put in their child's or the person they're taking care of, how many put in their drug information? When you distill it all down in that way, you end up coming down with a rather small number of 300 people. But still useful because this represents a much broader spectrum of people than we first looked at, who's being cared for by many different physicians and with many different treatment practices. Here's their demographics. So we had 300 people. The average age of them in beginning to use these tool was about 19 years. So this is skewing more towards children and young adults than our prior study. We don't have detailed information on their diagnoses for the most part, and that's very much dependent on the parents putting that into the tool, and most of them did not. So we don't have detailed information on most of that. Here's the drugs they were exposed to. And this is the drug exposures of all of them in the database, regardless of whether they entered into our analysis or not. But you can see somewhat similar to what I showed you earlier, but skewing a little bit more towards some of the more recent drugs, because in our prior data set, that went back 30 years, so a lot of those were phenytoin and phenobarb and so forth. But we see levetiracetam or Keppra, lamotrigine, valproate, clobazam, very prominently represented there, lacosamide or Vimpat. So a little bit different representation. Our patients, 303 patients that we analyzed, on the average, for the purposes of our analysis, they were exposed to 2.5 different drug regimens. And this is over relatively short time of the data that we analyzed. Their average seizure frequency per month was pretty high, 9.5. So this is a pretty refractory patient population. If we compare it to our prior population, we can see that the seizure tracker data set, we had more patients, 302. They were younger, but we had a lot shorter period of data collection time. And so if you multiply the number of patients times the number of years of data, you can see that in our prior study, we had about 3 times as much data as in this study. And in our prior study, our patients on the average had been exposed to 6 different regimens over time. That's because we followed them for 17 years. Here we had 2 different drug exposures on the average. Okay, so let me get to the one slide that's the punchline from the seizure tracker study. And the punchline is that, kind of similar to our FurCrest study, most of these different drug regimens were really kind of lost in the wash in terms of showing anything that was better than anything else. So our top drug exposure was levetiracetam in monotherapy, and really it was not at all demonstrably superior to anything else that people had been on. And you can go down the list there, lamotrigine, valproate, both in monotherapy, and it's really nothing is really emerging to stand out. But our one drug combination that emerged from the prior study, lamotrigine and valproate, in our study, only 12 patients had ever been exposed to it. Now, there's a reason for that, because it's actually kind of difficult to use these 2 drugs together. They interact with each other. Valproate drives up the lamotrigine blood levels. People worry about that causing a rash. So clinicians often kind of shy away from using these 2 drugs together, to be perfectly honest. But if you look in the fine print there, of the 12 people who were exposed to it, on the average, they had a 40% decrease in seizure frequency. And that was the largest change in seizure frequency of any of the drug exposures in this study. Now, statistically significant? No, because only 12 people. So too much noise in that sample to say, yes, this really meets our standards of this was something significant to be worth talking about. What are the issues with doing things this way? Well, one issue is that we find that parents will engage with the seizure tracker tool sporadically. They'll put in seizures for a while, and then whatever, life gets busy, or they lose interest, and then they don't for months and months. And so you kind of look at that. When you look at the data, it looks kind of like it's spotty. And so it's a challenge to figure out how to analyze it. What was going on in that interim period? Were they seizure-free? Well, probably not. But what was happening exactly? So that becomes a little difficult. Another issue is that we just, there's the way these tools are constructed at present, and they're evolving, but the way they're constructed at present, we don't have a lot of what we clinicians would consider hardcore clinical data. What was the EEG? What did the EEG show? What's the genetic diagnosis? And hopefully, as the tools evolve, there will be ways for parents and physicians even to put that stuff into the tool, which would help our analysis quite a bit. So, what have I told you? As I started out saying, I'm not trying to present our findings as this is the answer to your children with Lennox-Gastaut. That is certainly an unproven proposition. So don't go running out to your physicians tomorrow and say, you've got to be on this drug, my child has to be on this drug combination, because I just heard this guy from the University of Washington, and that's a pretty good place, and he said, so don't do that. This is always an individualized discussion, and by no means is the data, the gold standard of this kind of data would be a prospective, you get randomized people, you try them on something. The kind of trials that frankly have been missing a lot in the epilepsy therapeutic world is that, prospective kind of trial. And maybe there will be maybe there will be one of those for lamotrigine and valproate together. But I do think this is telling us something. We saw one drug combination that stood out in two very different populations, ascertained with two different ways, and to my mind that means something, and it's worth thinking about. We are going to go forward with this method of analysis. We have an agreement with the Epilepsy Foundation to acquire their data, and they have a tool just like Seizure Tracker. It's called My Seizure Diary. Anybody use My Seizure Diary? Okay, not that many. They actually have about twice as much data, or maybe more, than Seizure Tracker, and we have recently signed an agreement with them to acquire their data. So I think if we combine the My Seizure Diary and the Seizure Tracker data together and repeat this analysis, we will probably, if this drug combination is effective, or some other drug combination, I think something will bubble to the surface of meeting our statistical significance tests. And we may reach out to the LGS Foundation to see if they would like to support that effort further. Now, what could you do to support this? You could sign up for one of these two software tools and start putting your data in there. The LGS community as a whole, we need treatment data that's specific to LGS. And I can't assume that anything I've told you is specific. But if you're out there putting your data in, labeled as here's my child with LGS and here's how they responded to treatment, then that's going to over time add up to something. And that's something you can do with your sweat equity, but it doesn't cost anything. And so I think it would greatly advance the field to be able to have that kind of information. The last thing I want to just say on a basic science point of view is I told you about this pharmacodynamic synergy, which is just a way of saying we think these two drugs are doing something at a cellular level that is unique. And if we understood what that was at a basic science level, then we could, I think, craft a drug discovery process that's not relying on just kind of combining stuff that's already out there, but really targeting a mechanism that might be even more effective. And so in my mind, that's a very important way to go forward at some point in the future. Let me just acknowledge the grant support over the last 10 years that has come for this, our work, University of Washington, the Epilepsy Foundation, some industry support from UCB Pharma, and your own foundation. The people who helped us do this research actually, I relied a lot on undergraduate labor to kind of collate a lot of this data. Lindsay Warner and Christina Castagna, both whom are medical students at this point, and also our people at the two sites of the Washington State Institution. And also Rob Moss, who generously helped us with the data set, providing it to us. So here's one of my students scaling Mount Rainier. I wish I could say it was me. It's not. But sometimes doing research and treating people feels like this. You're just kind of slogging step by step, getting a little hypoxic, getting tired. But then, then the sun comes up and you look around and you see something amazing. So thank you for your time. I appreciate it. Happy to take any questions.